Go.
Hello everyone and welcome to today's lesson. We're going to be talking about aseptic technique. Septic means to be infected by microorganisms, normally ones that are pathogenic or harmful. Aseptic means without those microorganisms. So what we're trying to do is we're trying to create a work area that is without microbes. And the first thing in our effort to remove microbes from this area is the Bunsen burner. What the Bunsen burner does is it sets up a convection current. Effectively, it's heating up the air is then rising and then falling away and being replaced by new air. In doing that, it's creating a bubble around this area where microbes are unable to land on the stuff because they're being sucked up and away from the area. So that's quite a useful thing for the Bunsen. Our Bunsen is also going to be our primary sterilisation technique. And sterilisation just means to kill off the microbes. So flame is going to kill off most things. So before I started, the thing I've done is I've actually washed my hands to make sure I've got most of my microbes off there as an extra precaution, and this area has been sterilised down with a bit of Virkon to kill off the microorganisms. So first things first, we have got some agar plates. Agar plates are what we're going to be growing our microbes on. What they are is a petri dish, and you might be able to see there is a layer of agar jelly in there. Agar jelly is a nutrient jelly. It contains a source of carbon or sugar, and it contains some of the other nutrients that the bacteria will need or the fungi will need to grow. So that's what we're going to be growing in there. We're going to keep this within about 30 centimetres or so of our Bunsen to keep it sterile or aseptic. And what I'm not going to do is I'm not going to open this because that invites the opportunity for bacteria to land. Even with my aseptic bubble, keep it closed as long as possible. Okay, so I'm going to start off by sterilising the tools I'm going to be using. So let's say I was going to be using something called an inoculation loop. That's this piece of equipment here. It's got a little loop on the end, it's basically a bit of wire. And the first thing I'm going to do is I'm going to pass it into here and I'm going to sterilise it with heat. That will kill off any bacteria, any fungi, any spores on there. That's now sterilised. I'll just let it cool down in the air. What I don't want to do is take this and let it cool down on the desk. Why? It's just giving it the opportunity for more bacteria to be landing on it or to pick it up to the surface. So that's now possibly septic, so we need to sterilise it again. Okay, so that's now ready for use. And what I would do is I would take that, keeping it near here, I would take my bacterial culture. In this case, I've got some E. coli. This is a relatively safe bacteria to grow. And I could then put that in here. And I've now got some on there. Put the lid back on. And then I can spread it onto my plate. Now I'm going to keep it open only as long as I need. And I would do what we call a smear. So I would smear it along here and then close it immediately. That's now ready to go, and I would sterilise this again. There's one final thing I would need to do with this, and what I would do is I'd take some tape and I would seal this, and that would again prevent it from being opened unless I wanted it to be opened. So I could then put that into a incubator, incubate at a warm temperature, and see what microbes grow. So that's one way I could use my inoculation loop. What about if I wanted to make an even carpet of bacteria all the way across one of these? Well, I'm going to need two bits of equipment. I'm going to need a pipette, which again I'll just pass through there to sterilise it, and I'll sterilise it again when I use it. And the other piece I would need is a spreader. So to sterilise the spreader, I could pass it through there, but I'm going to show you another method you could also do. That method involves a bit of ethanol. So pour some ethanol into there. Ethanol is flammable, and I can put this into here, give it a good soaking, and then I can light that on fire and just sterilise it. That's now sterilised and ready to be used. Again, I'm going to put it down just because I'm going to show you the rest of it. So, step one, I want to make a lawn of bacteria, an even thing. Sterilise my pipette. That needs to be in there for a fraction of a second because it won't melt if it's not too long. Open up my petri dish. open up my bacteria, take a small amount of my bacteria onto there. That should be enough. One more drop, there we go. Okay. Now I actually made a little mistake there. And my mistake was I accidentally squeezed a little bit back in. So I could have contaminated that. So that's something to bear in mind. Okay, so now I'm going to sterilise my spreader. Get back in here. Light it on fire. Let it burn down, give it a second. And now I'm going to spread it around. So I'll use that as a spreader. And I'm being really gentle here so as I don't break the surface of the jelly. Close it up. I'd then secure it with my tape again, and I would put anything I'd need to write on the front. So I'm going to put my name, I'm going to put the bacteria, E. coli, and I'm going to say it's a lawn because I've made an even spread, and I'm going to put the date that I've done this at. So when I put it into the incubator, I know when I've done it. So that's how you do aseptic technique. It's Mr. Robinson signing off.